Around 44 million caregivers provide care to someone who is ill, disabled, or aged. Due to this environment of chronic stress, their own health often deteriorates. Our hope is to identify targets to alleviate such stress. Inborn errors of metabolism, or IEM, are conditions associated with a particularly heavy caregiving burden, as such individuals are always at risk of plunging into metabolic crisis. However, few studies have looked at the parents of IEM children and studied their stress using a biopsychosocial model, which I plan to tackle. I focus on the neuropeptide arginine vasopressin, or arginine. Arginine plays a crucial role in stress as it can stimulate cortisol secretion. Now, cortisol is the primary stress hormone released by the body. Under chronic stress, cortisol is excessively secreted, and this puts one at risk for increased vulnerability to dozens of diseases. Arginine is also linked to negative coping strategies. As shown in the model, I hypothesized that parents of IEM children versus parents of healthy children will self-report different levels of burden, and that this will be associated with different levels of arginine, and finally, that the use of certain coping strategies can moderate this relationship. The results show that, indeed, parents of IEM children reported higher levels of burden and expressed higher levels of arginine. Furthermore, the interaction between the use of positive coping strategies and burden was significantly associated with arginine levels. For those not using any positive coping strategies, as burden increases, arginine skyrockets, as shown in the dotted line. The solid line represents individuals making an average use, and of greatest interest to us is the dashed line. So for individuals making tremendous use of positive coping strategies, even as burden increases, arginine remains at a very low level and in fact slightly decreases. This has important biological implications because if the use of positive coping strategies can decrease arginine levels, this means that you may be able to decrease the excess secretion of cortisol and its downstream negative health effects. In future research, I plan to study the biomarker oxytocin. Due to the great structural similarity between these two molecules, researchers have actually shown that oxytocin can bind to arginine receptor in vitro, and they speculate that such binding is determined by perceived emotional context. This begs the question, is the response of arginine due to arginine binding to its receptor or due to the binding of oxytocin? By analyzing oxytocin levels as well as our interview transcripts, we will better understand the psychological influences behind the biological stress response. We will gain deeper insight into targets that can perhaps be used to screen caregivers at risk of vulnerability to disease, and we can help caregivers best take care of their loved ones while also taking care of themselves.